So welcome to the second session of the 2022 Arbor Day webinar series hosted by the Rice Creek Field Station and the Canal Forest Restoration Project. If you didn't meet me last week, my name is Robert Salerno. I am the Canal Forest Restoration Project intern, and I'm joined by Dr. Kristen Haynes, who is the Assistant Director of the Field Station, as well as one of the directors for the Canal Forest Restoration Project. And so today we have another great talk lined up from Dr. Gregory McGee, who is an associate professor at SUNY ESF and the director of the Wildflower Restoration Project there. Dr. McGee has current interests in forest and ecosystem processes and forest and ecosystem health, which really ties greatly into the promising restoration practices being developed for wildflower restoration. And today we'll be able to hear what us citizen scientists can do to help this cause. And so before we get started, just a couple of housekeeping things. If you're not the person talking, please remember to mute yourself. And if you have any questions, feel free to put them in the chat. We'll answer those all together at the end, or if you'd like to hold on to your question, feel free to ask it then. We will be recording every talk and they will be posted to our YouTube channel. Our talk from last week from Carolyn Merschisner with the New York State Hemlock Initiative, her talk has already been posted to our YouTube channel. So if you missed that one, please feel free to check that out at your leisure. This one should be posted within the next week. And don't forget that we do have 3 more Fridays in April with talks on each of those Fridays, same time, same place. So if you enjoyed this talk and the others, we look forward to seeing you there. And I think that's all I have to say. So Dr. McGee, you can go ahead.
Okay, Robert, thank you. Robert, Kristen, thank you for the invitation to join you this afternoon and participate in the seminar series, and good afternoon to everybody out there. I'm going to see if I can talk and navigate my screen right now and share the screen while I'm going. It's difficult for me to do that sometimes. So I have maybe just a discussion I'd like to share with you in the context of this seminar series. My understanding is all of you have an interest in forest restoration and forest reforestation. And oftentimes that focus is the focus on reestablishing tree cover and forest cover. And I'd like to come with you with a little bit of a perspective about some of the other plant species that we ought to be concerned about. And so I'd like to couch my thoughts about this, maybe in a springtime, thoughtful and hopeful sunny day, in which not too much of a distant future, you're going to be able to go out and perhaps find a forest that looks like this. And if you're anything like me, you might need to look around and check around and find a few places before you can locate a forest floor that has such a riotous display of wildflowers. And so what I'd like to do is discuss with you as students, maybe as some woodland owners, perhaps as some interested volunteers, some things to think about in terms of how and whether or not, and in fact, the need for restoring herbaceous understories within the New York landscape. And the way I like to think about this is this type of display, frankly, I know is quite remarkable for most people. And for many of you, you have a little haunt that you like to go to find this type of abundance of wildflowers. But quite frankly, this should be unremarkable. You should not have to go out of your way to find this. And so part of what I want to discuss is to maybe lay out some context and background for maybe a biodiversity and conservation issue that we're facing these days in the Northeast and the temperate zone, let alone in central New York, and then talk about some ways that we can think about restoring these understory communities. So before I get going, I would like to thank a few people, because sometimes I get ahead of myself and forget to thank all of the folks who have been involved in this research. This has been going on for a few years. The McIntyre-Stennis Cooperative Forestry Research Program has supported this work. I'm going to be speaking about a couple of student projects that have been conducted over the past years. And so those student projects have been supported in part by some very generous donations of foundation money from the Garden Club of America. The National Fish and Wildlife Foundation Native Plant Conservation Initiative has supported this work, along with the Biodiversity Research Institute in Albany. All of this work has been done on state land and private land. There are too many individual landowners to thank for this, but there are so many folks across New York have an interest in this. I want to thank all of them, and certainly as well as my colleagues and collaborators at Lands and Forests at DEC. So getting back to the question of restoring unremarkable herbaceous communities, this is where I'd like to go. I'd like to share with you my perspective of the extent and the magnitude of this problem of depleted herbaceous understories in New York. I have some perspective to share with you all on that. I'd like to then share with you some investigations about how we're teasing apart what the consequences and causes of this issue are. Are we looking at issues of dispersal limitations by plants? Are there matters of pollination inhibition that we need to be concerned of, and maybe we don't have a lot of control over that? Another factor that we might have some control over is light. I want to share with you also some results of some pilot restoration projects, maybe as some motivation or as a how-to with all of you, maybe to restore some woodlands along Rice Creek or other places within Oswego County and elsewhere. Okay, so that's where I'm going to go with this conversation. Let me get started, maybe just with a landscape that I suspect everybody on the call is familiar with, right? This is a landscape that could have been taken anywhere in upstate New York or other northeastern states. This is my backyard. I happen to be north of Skaneateles, and this landscape that's fragmented and divided by agriculture with a variety of woodlands throughout is something that we're all familiar with. And so the habitat, the natural habitat and the conservation need for many of these plants and the organisms that rely on these plants are habitats that are scattered within this matrix of agriculture. And if we burrow into that view a little bit and increase our zoom and resolution, I think we're going to see what many of you might be familiar with if you've stared at aerial photographs in the past. And that is that you can see a change in texture within this forested block here. We can see evidence of old fields with the tree lines that still stand out against the remaining trees. Those tree lines, maybe 40, 50 years ago, were the tree lines that bounded some of these open fields. And you can see that these fields are shrubbing in. In the day, it probably would have been dogwood and elderberry and viburnums. These days, I'm sorry to say, this is not going to be the point of the talk, but now it's buckthorn and honeysuckle and privet. But through time, these forests, these shrublands would then grow into closed canopy forests. And so you can see over here, this is going to be the woodlot that's going to be the focus of some of our discussions. This woodlot is about 80, 90 years old. It's closed canopy forest. It has mature trees on it. And you can see just from the texture of these trees that these trees tend to be small in crowns. They've grown under competition. They're even aged, but there is closed canopy forest here. And you can compare this forest of closed canopy, this even aged forest, with a forest that's adjacent to it. And this is a remnant forest that had never been cleared for agriculture and has much larger trees in it. And you can see the evidence of the gaps that are between these trees. So larger trees with gaps, and you can see the one hill slope going down to the swamp also has a number of large trees on it with gaps in it. And I want to talk about the evidence of this past land use, not only from the air, but also from the ground. And if we go to this place and compare, say, this tree line that had been the edge of a field back in 1938 with the same tree line, the picture was taken in 2015, and we look around, we would see things like this. I suspect many of you have seen this in woods in your neck of the woods. A stone wall running down through the middle of the woods. And so what this is evidence of cultivation, evidence that people were picking the stones, putting at the edge of the field. And you can find this evidence today. And so this location here would have a stone wall. I'll show you that specific stone wall in a moment. Interestingly, and just as an aside, this was new to me. I don't know how many of you have ever seen this. And by the way, I'm losing track of folks now with the way my screen is showing. If any of you have a question, I'm going to invite you to pipe up, interrupt me, because I think I'm not going to maybe see you if you raise your hand. I'm going to invite questions and conversation as we go. So please don't feel the need to hold your questions to the end. And one of the questions I would have for you is if anybody has ever seen anything like this, a row of stumps lined up in the middle of the woods. And where I needed to go to find this, the first time I ever saw this was down in the Southern Finger Lakes. Has anybody seen this? I'll give it a moment for anyone to just holler and mic up and yell yes. Maybe no. So this is evidence of some past agricultural use. And what you're seeing here are white pine stumps that were quite literally torqued out of the ground. Whenever this area was cleared in the Southern Finger Lakes on the shallow clay soils, pine was a very dominant tree. So the area was cleared, a field was cleared, and what the settlers would do is link up a team of oxen and walk them in circles until they just broke this stump out of the ground. They then dragged those stumps to the edge of the field. They would be 8-10 feet tall with the entire root systems on them. And they used those as fences. They're fences that were essentially made of stumps. And 100 years later, you can still see evidence of that activity through the forests. Just another example, this is a state forest on the south end of Skinny Atlas Lake. This is Bear Swamp. I have cored some of the trees in these old fields. These are better than 120 years old. So 120 years after the fact, you still see remnants of the stone walls throughout the fields and consequences for the understory vegetation and diversity. So if you go to these stone walls within the forest and you look one way, you would see a canopy that's maybe broken up and uneven age with some shafts of light coming down through it. And if you turn the other way into the old field, what you would see is these smaller but mature trees, all of them having smaller crowns. And so what you're seeing on the ground is that evidence that we saw from the aerial photographs a few minutes ago of the texture of those forests. Going back to that specific place where I started this conversation, if we go find this tree line and the stone wall, this is a picture of that looking to the east, right the way the arrow is pointing in that upper picture. And if I look now to the south into that old residual woodlot, we have a riotous display of trillium and false Solomon seal and leek and blue cohosh and a whole litany of native understory plants. If we turn 180 degrees and look right into the new growing successional forest to the north, that's what we see. Closed canopy forest, a lot of trees, so the ecosystem is reclaiming itself. This is good. We have carbon sequestration, we have control on the hydraulic cycle. There is habitat that's being provided for wildlife, but those wildflowers are largely missing on that forest floor. So while the forest is established, the wildflowers have not recolonized. And this has happened in this particular place after about 80 or so years of regrowth. Those flowers still are not past that old stone wall. So I want to give you a little bit more information, maybe broadly, paint this with a broader pen in terms of knowing and understanding the consequences of this across New York State. So if you dive into the literature, maybe some of you are aware of this literature, people have been recognizing this phenomenon of post-agricultural depletion of understory native plants in the Northeast, probably for about 30 years now. A lot of those studies were focused on one location. They were case studies, or maybe a couple of spots within a county or so. My interest a few years ago was to go across New York and document this phenomenon across the different geographic regions of New York. So we went into the northeastern highlands of the Adirondacks, the Tug Hill, and the mountains east of the Hudson River. We looked through the eastern Great Lakes lowlands, and then also into the southern tier and the northern Appalachian Plateau. And the differences within these zones, frankly, it's not just climate, it's also soil conditions. And this demarcation, this is going to come up a little bit later. The demarcation between the northern Appalachian Plateau and the northeastern highlands into the Great Lakes lake plain has to do with soil conditions and parent material. Everything's been glaciated through here, but the soils to the south are glaciated on acid sandstones and shales, and the soils development your way through Oswego is a little bit more due to development on limestone and more base-rich parent material. So there's issues of soil texture and chemistry that come up when we look at plants across New York. So let me start here by sharing with you just some results of this study to lay out some of these problems with the plant diversity. First of all, just a little bit of a study design. I went to these different locations. There were 33 sites, and at each of these sites, these represent paired residual forests with forests that were regrowing following succession after agricultural abandonment. And so in this schematic, what you see, this bold line here would be that stone wall between the residual forest and the secondary forest. And in parallel lines along this stone wall, I documented the plants that were present in that residual stand with 2 transects at -5 and 10 meters away from the stone wall. And then I placed transects 5, 10, 25, and 50 meters into the secondary woodland. And along those transects, I set up quadrats. They were 1 square meter quadrats, and we didn't measure cover. We just measured the presence and absence of different species. So the data that we're going to be looking at are frequencies of occurrences of different types of vascular plants at different distances away from the stone wall. All right, so let me show you the outcome of what we found here. And I don't want to dive into these 2 graphs too much. What you see here are 4 panels with different species listed on them that all show similar trends. There's meaning to the reason we have these 4 panels. One reason I wanted to have 4 panels was just to not have a muddle of lines, just to be able to show these different patterns. We'll come back to these 4 panels because these species are grouped by dispersal mechanism. So we'll see this in a few more minutes. But what I want you to see here, the data on the y-axis are relative frequencies. That is, along that transect of 17 quadrats placed along those different transects at these different distances away from the stone wall. The stone wall would be distance 0. So at 5, 10, 25, and 50 meters, this is showing the frequency with which different plants are established at different distances away from the stone wall. And so what you see here very clearly is that within 5 or 10 steps away from that old ecotone, many of these plants, many of these native plants just drop down to about 10, 5, 20% of their local abundance. So many of these plants, while they're present and abundant next door, diminish down to nearly 0 within 50 meters away from those stone walls. All right, so we have some consequences of this past agricultural use. For some reason, some way, these plants are not establishing quickly and readily within these successional forests. Okay, so let me show these data with you a little bit differently, and this might require a little bit of explanation. Give me a moment to maybe step through this, this display with you. This is an ordination, and an ordination is simply a way of displaying similarities and differences between observations. So what you see here are 66 squares. Those 66 squares represent the 2 sites, the 2 paired sites at each of the 33 study sites across New York. And very simply, excuse me, I'm sorry, the way you interpret this display is to just understand that compositionally, 2 points that are plotted close to one another on this ordination display are compositionally similar. The farther they are along this display, the more dissimilar they are. So you would interpret this display as saying, well, okay, these 2 points represent the composition, the vascular plant composition of 2 forest stands across New York, and they seem to be very similar to one another. Likewise, these 2 points here are very similar to one another. And in fact, they're as similar to each other as these 2 are to each other. But these 2 observations are quite dissimilar from these observations. So there are trends, and there are changes maybe due to land use and due to environmental conditions that influence the distribution of these points on this display. So right now, this display gives you no information. But I have information about what's going on at each of these 66 points. So I can begin interpreting this display by putting some additional layers onto this. So for instance, my computer knows which of these points are residual forest stands and which are the post-agricultural secondary stands. And if I light them up with different colors, what we can begin to see is some pattern. And so although there's some overlap in the composition, the tendency with these data are that the residual sites tend to plot to the upper side of this diagram, and the secondary sites tend to plot to the lower side of the diagram. And if we want to apply some statistics and some hypothesis testing statistics, we can see that there is a significant difference, a highly significant difference in the ability to determine secondary forest understory composition from primary forest understory composition. We can add more information to this. At each of these locations, I took some soil samples and measured the soil pH. And maybe you knew going into this, as I was showing you that we sampled across these geographic zones, that there would be differences in soil condition. So the other thing that's happening here is that along this x-axis, this horizontal axis, what we see is a gradient of pH with more acid soils in the Adirondacks and the Tug Hill and the southern tier. As we get toward the lower elevations and toward the lake plain, we see soils that have higher pH. So the composition differs not just on the land use history, but also on the soil conditions. So it matters in terms of what you're restoring to know the types of soils that you're working with. So just as I can make this topographic map of pH, I can also make a topographic map of the abundance of these different plant species. So I'm not going to muddle this map with these different topographic maps for different species, but what I am going to show you is maybe the high point, the peak, so to speak, of a topographic map for the abundance of different plant species. So those peaks are shown here, all right? So what this is saying is that along this ordination display, trout lily acquired its highest abundance or its highest frequency at this point in that ordination display. Painted trillium was over here. The other 2 trilliums, red and white trillium, were on this part of the display. And so what you can see are these species that we were able to discern as indicators of residual forest lands. That is, they do not occur with great abundance in the secondary forests. They all light up on the top side of the display. Those species that characterize the secondary successional forest are species like speedwell and avens and dogbane and the various club mosses. And they are lighting up at the bottom and the right of the display. All right. So this is a long way of saying that the composition of these woodlands differs between residual and secondary woodlots. Let me tell you why I think you should be concerned about this. I think it matters. It's one person's opinion, but let me tell you this one person's opinion. Just a picture of these different species, just so you have an idea of what they are, and perhaps you're familiar with many of these. But if you look at the landscape across New York, and you look at trends in the abundance of forest cover through time, from European colonization in the 1600s until just about present day, and you look at forest cover, This top line shows trends for the northeastern states, and the bottom line shows the trend for New York. It's a similar trend. It's a sort of a parallel line. We don't know what the pattern was in between the 1600s and the beginning or the late part of the 19th century. But we know that when we started keeping these types of records, that by the 1870s, 1880s, we had gone from about 28 million acres of forest cover in New York down to about 7 million acres. Since that time, since the Industrial Revolution, since the onset of the Great Depression, when people had to up and leave their farms and default on their mortgages and they left, there's been a lot of forest recovery within New York and the Northeast, and we're back to about 18 million acres. So the question might be, well, what does 8 million acres, 7 million acres of forest look like? Well, if you go to the Adirondacks, that's about 6 million. The Catskills are less than 1 million acres. And I'd like to direct you to not the entire Tug Hill, but the central core of the Tug Hill. These are all landscapes that were never widely cleared. There has been clearing, there has been cultivation, but if you add up the amount of forest cover in those different landscapes, yeah, that adds up to about 7 million acres. So here's the take-home message. If there were 7 million acres of forest in New York at the turn of the 20th century, and if we have gained back another 10 million acres, well, those 10 million acres of new successional forest are scattered throughout New York, but probably not in the Catskills, not in the Adirondacks, not in the central core of the Tug Hill. They're everything else. Frankly, just about the vast majority of all the forest outside those landscapes are secondary woodlots. That's perhaps, if you're in the mood for going out to find trillium these days, you might need to have a destination woodlot to go find that trillium. Just another way to beat this dead horse for you. In Onondaga County, what we know from this type of aerial photograph analysis is that in 1855, there were about 23% of the total land was forested. At the time of the Depression, that was below 10%. By 1980, it had come back to 40%. So what this means is that in Onondaga County and in Oswego County and other northern and central New York counties, the forest land that we see out there, if we went from 10% cover to 40% cover, that means that better than 75% of the forest cover locally is post-agricultural second growth woodland that doesn't have this vast diversity of understory wildland plants in it. They're depauperate of this woodland diversity. So what do we do about this? The issue is, what are the causes of this slow recovery of these plants? Is there anything that we need to learn in these lessons of what might be causing this? And then how do we restore these native assemblages? If you're interested in doing this in some of the woodlands at Rice Creek, if you're interested in doing this, if I have some woodland owners on the line, how do you go about doing this? Is there evidence that this can be successful? So let me walk through some additional research here. The kinds of things that we've been looking at in my lab for the last several years is to look at matters of dispersal. Are there any dispersal limitations? If these seeds are dispersing, is there something that's inhibiting their establishment? Is there something about the soils after maybe a century or more of cultivation that causes these plants to not be able to establish? If organic material is rebuilding, maybe there's mycorrhizal dysfunctions that are not allowing these plants to establish and get the mineral nutrients that they need. There are other matters of pollination, right? So we know that there is concerns about pollinators globally. Is there a lack of pollination services that's keeping these plants from even undergoing sexual reproduction so that we can have the seeds to disperse back onto these secondary woodlots? And then on top of this, you have to always keep in mind these plants might be dispersing, they might be establishing, there might be pollination occurring, but once those plants get established, the white-tailed deer come along and knock them right back down again. So no matter what you do, if you don't control the deer, we might not have success with this restoration effort. So let me walk you through some of these these approaches. I'd like to introduce a graduate student who is a recent grad. The person in the background here is Jeff Griffiths. He did his master's with me a few years back, and he had an interest in this pollination question. And there's two sides to this work. One side of it is does the lack of herbaceous diversity in these secondary woodlots impact pollinators? That is, are pollinator populations influenced by the presence or the absence or the abundance of these different diverse plants? The flip side of that is there a lack of pollination service that would impact the ability of these plants to recover? So Jeff had a few aspects to his research. Briefly, what he did is he went out and he put pan traps out of different colors, white, blue, and yellow, in an array at several sites. And so these are some sites across Cortland, Onondaga, and Oswego County where he worked. And he gathered information about pollinators that made a wayward turn into his soapy water. And he was able to assemble an understanding of the composition of the pollinator systems within these forests. There's going to be a little bit of jargon coming up here momentarily. This red line is the demarcation between that Appalachian Highland region and the Great Lakes Lowland region. So what he did is he set his plots out spanning these regions to make sure that he was looking at different forest types within this study. All right, so that's going to be important here in a moment. So real quickly, I don't want to get stuck on this, but it was interesting to us, his outcome. What these two graphs are showing is the bee abundance, that is the number of bees per site on average, and the bee species richness on average per site. In the eastern Great Lakes lowlands, these two bars are the low elevation sites, and the northern Appalachian Plateau, that is the southern tier sites. All right. So what you're seeing here is that there's a difference between the forest type, but interesting to us, what we were really expecting to see, we thought this was going to be a slam dunk. If there are no flowers out there for pollinators to pollinate, when you go sample pollinators, you should probably find fewer pollinators in the secondary forest versus the residual forest. So the 1 and the 2 is referring to primary residual, and the 2 is the secondary forest. So in both the eastern Great Lakes lowlands and at the Appalachian Plateau sites, in all of these cases, there was no difference between the residual and the secondary forest, which was really interesting. It was an interesting outcome to me. So regionally, there's differences, but locally, based upon land use, there didn't seem to be an effect of a lack of these herbaceous plants for maintaining populations and diversity of pollinators. The flip side to this question was whether or not the pollinators themselves influence the ability of these plants to apparently reproduce. So Jeff set about conducting a pollinator exclusion experiment. And in one treatment that I'm going to show you, what he did is he packaged the inflorescence of various species with this mesh net, put a tie around it in order to keep the insects, the pollinators, away from those plants. So what this will show us is whether or not plants can successfully self-pollinate, and whether or not having pollinators limits their ability to pollinate. The other treatment was a supplemental pollination. So what he did then was he distinctly took plants and very meticulously took the pollen off of the stamens of one plant and put it on the pistil of other plants. So he served as the pollinator, and he made sure that each and every pistil of each and every flower on each and every one of his experimental plants was pollinated. And so what he could do with this was, by just looking at open pollinated flowers, he could determine whether or not there is sufficient, effective, efficient pollination of the local pollinator bees in their ability to pollinate the flowers. So let me show you the outcome of this study. And we'll just take these panels in turn. And what I want you to see here for anemone, this is the liver leaf. What you see here between the exclosure treatment, the open treatment, this is just business as usual, just a flower that is open to bee pollination. And then Jeff's supplemental treatment. If you look at the exclusion versus open treatments, what this is showing us is that anemone, in fact, it can self-pollinate, but it tends to do better when there are pollinator services available to it. It can cross with itself, but it also does better whenever there are pollinators. But what we see with the anemone is that between the open and supplemental, it seems like the pollinator populations were sufficient across his sites to pollinate these plants as effectively as he could do it individually and purposely to make sure they were all pollinated. Same thing with allium. This is wild leek. With open and supplemental, we saw no difference in the way that these plants were pollinated. The pollinators seem to be doing a sufficient job. All right, what we saw with white baneberry is that, well, for some reason, white baneberry doesn't do any better in attracting pollinators for pollinating its flowers. And in fact, we could find no difference between the exclusion treatment and the open treatment in terms of this outcome. And I should give you a definition of what this y-axis is. What he measured was the seed to flower ratio. That is, how many seeds were there per flower. If there was better pollination, more effective and more efficient pollination, then per flower at the end of the growing season, you would expect to see more seeds there. Myanthemum racemosum is false Solomon seal. And I don't know if you've ever had the occasion to squeeze open the berry of a false Solomon seal, but there tends to be one seed per berry. Sometimes you might see two, but usually there's just one. So we're not seeing any effect from myanthemum, but I sort of think it was because of how we chose to measure this. We weren't going to be able to get many more seeds in myanthemum anyway, because it doesn't tend to produce more seeds than just one in it. But the take-home message from this is that it seems like the pollinator services are being effective and efficient, at least within the sites that we've been working with. It doesn't seem like pollination is limiting these plants to reproduce and to set seed to be available for reestablishing on these sites. Now, another part of this story is that Jeff then wanted to know if you can imagine in these post-agricultural sites, maybe a lucky seed would get dispersed out 50 meters out and then grows and has the ability to establish and begin to flower. Jeff's question was, well, do the bees find those flowers if they're off on their own? Is the search image there? Is there a difference? Is there an interaction, say, of a wild leek, Allium tricoccum, or this anemone, the liver leaf? Is there an interaction of those plants growing next to other plants like them that are all flowering, and collectively that display of flowers would attract pollinators, versus a flower that gets established out in the middle of one of these empty forests might not be able to attract the pollinators? So that's what this study did. And what Jeff did was took flowers, put them in pots, made sure that they were blooming. And during their blooming phase, he set them out 50 meters away from a residual woodlot and then set other ones in the woodlot. And the outcome of that is shown here. So for allium, the wild leek, there seemed to be no difference in the ability of pollinators to find this the allium out in the middle of the post-agricultural forest on the right versus the residual forest on the left. But when we look at liverleaf, there was a difference. For the liverleaf, those plants that were placed in the residual forest had about 20%, 25% more seeds per flower than the same plants that were planted in the post-agricultural forest. So this is something we need to go back to. This is intriguing to me. One of the things I'm wondering about is with the abundance of pollinators midsummer when leek is blooming, maybe there's no lack of pollinators to find these. Anemone can bloom underneath the snow. It might be blooming right now in your neck of the woods. I'm not quite certain of that. Not quite here, but it's pretty close. So it might be that early on, whenever things are still cold and the insects are not quite active yet, that they're not finding the anemone as easily out in the middle of the post-agricultural forest versus the residual forest. So the point here is that there might be some evidence that these plants may not actually get the pollinator services out in the middle of the woodlands. It might depend on species, and for me, this is a follow-up study that we need to get more information about. Dispersal. All right, so this is that same graph that I showed you before. Now I've just given you some information about the mode of dispersal. These are graphed and grouped by the way in which their seeds and fruits get around. An anemocor is a plant that disperses by wind. And frankly, it's interesting to me that any plant cannot get established in these post-agricultural lands whenever their spores or their seeds are dispersed by wind, but in fact there are. Zoochores are plants that are dispersed by animals. Barochores are animals that are dispersed by well pressure. That is, they fall to the ground and there they sit. They don't have known dispersal mechanisms, except that, well, you know, we know that Corylus cornuta is hazelnut. So acorns, beech nuts, hazelnuts would be characterized as barochores. We all know that these are then dispersed, right? Animals will pick them up, they will disperse them. The issue with the terminology is, though, that the ecologists are looking at these barochores as the seeds are being predated. They're not being dispersed. But we all know that jays, birds, small mammals will cache these nuts and fruits sometimes and serve as dispersers by default, if and only if they forget where they put them. Nonetheless, we played nice and categorized our plants as barochores. Myrmecocores are plants that are dispersed by insects. So one of the things that we did was to see, are there differences in the ability of these different groups to disperse into these woodlands? And so the way we did this is, if you draw a best fit line and you characterize the slope of this logarithmic decay function, you can calculate a slope for these lines. So what we did is calculated the slopes of these dispersal patterns for each of these categories and then found the averages. So what we see here, actually, let me back up. A very steep slope would have a large negative value. More shallow slopes would have a less negative value. If the slopes, in fact, were positive, there would be a positive slope. And for those plants that were secondary successional indicators, like those club mosses, they in fact had curves that looked like this rather than having these negative slopes. And then a slope that showed no difference, that is, if a plant was equally abundant, no matter whether it was in the residual woodland or at different distances into the secondary woodland, it would have a zero slope, all right? So we found those averages, and what we see is that the anemocores, the wind dispersed, the non-dispersed, and the animal dispersed had many species that had positive slopes. They had many that had zero slopes. And when they did have negative slopes, they were not as negative as the myrmecocores. It seems like the myrmecocores, the ant-dispersed plants, are the ones that really have a difficult time making it into these secondary woodlots. So that got us wondering about whether or not the dispersal mechanisms are actually functioning within these myrmecocores. So enter the next student. This is Nathan Keel. Nathan was an undergraduate research student with me. He's now, by the way, he's pursuing his PhD at the University of Wisconsin. And he came up with an idea to test to see whether or not the ants were as active in the secondary forest at dispersing these myrmecocorous plant seeds as in the residual forest. So he set up a cafeteria experiment. And what he did is he set up trays, little petri dishes, in which he put 10 seeds of different species that we know to be myrmecocores: the liverleaf, the spring beauty, the bloodroot, and red trillium. And maybe before I get ahead of myself, I should mention to you that the reason that these plants are dispersed by ants is because if you look at a seed, this little appendage on the right here, this little creamy white appendage, is called an elaiosome. That elaiosome is very high in sugar and lipids, and especially the lipids. So the ants are interested in the seed, not because of the seed itself, but because of this little package of fat that's next to it. So what they do, they go gather the seed, carry it back to the nest, feed the fat to the workers and the larvae in the nest, and then they throw the seed out in the trash heap. They're dispersing the seeds and putting it in contact with mineral soil when they do that. So the experiment that Nathan set up was to put these trays out without any kind of inhibition. And then, so anything that's out there could feed upon these. Then he also put these trays in the middle of it's called tanglefoot. This is just sticky paper. It's fly paper, so to speak. So if an ant was trying to get to those seeds, it would get stuck on that paper, would not be able to get to the seeds. So this is a way to put the seeds out and control access for the ants. But you have to control for other things. There are other large and small mammals that might have an interest in feeding upon these seeds. So we needed to make sure that maybe apart from a wayward beagle, that perhaps we were keeping these seeds away from small mammals, mice and voles and the like. So we put some of these Petri plates in chicken wire. So this would allow the ants to disperse, to go into the cage, get the seed and walk out with it, but would keep the small mammals away. And then this setup had both the tanglefoot and the cage. So ideally, this would be a control that would keep everything away from consuming and taking these seeds. So his question was, is there a difference in the amount of seed removal within the residual woodland versus the secondary woodland? Are these ant communes actually functioning in the secondary woodland? So don't freak out at this graph. I'm going to walk you through here just momentarily. There are 4 different species. One of these graphs is not like the other. We're going to come back to this. Focus your attention on these 3 for now. And so what I want you to see is that for each of these, the treatment is essentially we gave access for all organisms to access the seeds. We gave ants access. This is just the cage. We gave rodents access. That is, this is the tanglefoot. It kept the ants out. And then we inhibited all these organisms from accessing these seeds. That's what the none treatment is. All right. This would be both the tanglefoot and the cage. The response variable here is the proportion of seeds that were removed. So a 1 would be all seeds have been removed. A 0 is all the seeds were still there 24 hours after Nathan put them out in the field. So the first take home about this is to recognize that our controls worked. These treatments that had the cage and the tanglefoot, there was nothing removed from it. He put 10 seeds out. He came back 24 hours later, 10 seeds were still there. Interestingly, the rodent access led to very low removal. This was interesting to us. We thought that the rodents were just going to predate these seeds and they'd be gone, but they didn't. Interestingly, we had some access to some other large regional data, and the year that Nathan did this was a year that overall small mammal populations had cycled to a low. So the small mammals just might not have been active in these forests to remove these seeds. All right. But what we do see is that for all of these plants, the ants were very effective at removing the seeds. All right. One point, this other plant, this the leatherleaf, we set this experiment up and started to work to gather the data. And then Nathan came to me and said, hey, you know what, Greg, I just read a paper. It turns out that the liver leaf has a false eliasone. It doesn't have any fat on it. So he was wondering if it was going to work out at all. Well, we didn't know that going in, but the ants sure knew that going in. They had no interest in taking the seeds of that liver leaf. So anyway, we removed that plant from our eventual analyses, but it was an interesting thing just to learn that by this particular mistake. All right, so the other part of this experiment then was to compare the residual forest to the post-agricultural forest. So the next graph I'm going to show you is actually the experiment. It is, was there a difference in the ability of ants to remove seeds but when you average between the residual stands and the post-ag stands? And so what he saw was, no, there was no difference. When we looked at just the ant removal, The ants were as active in the post-agricultural stands as in the residual stands. So they're there. It doesn't account for why we can't get these seeds into those woodlots, but it's not because the ants are not active. All right. Interesting observation. While he was doing this work, Nathan was seeing that there's an invasive slug that is predating on these seeds. This slug had an interest in the eliosome just the way the ants did, and it would come and feed on the eliosome, but it did nothing for us for moving the seeds around. It just left them in place. So an interesting observation while you're out there looking at things eating your seeds on the forest floor. Okay, so the seems to not be a issue of pollination. There seems to be no issues of dispersal. What else could be accounting for these differences? So all along this time that we're setting these experiments up, we've always been thinking, is there something about the conditions in those post-ag forests that just don't allow these seeds to establish? If you put them there, will they grow? So what we did for a couple of years was to get a bunch of undergraduate volunteers, went out and we had the students clear the leaf litter off the ground, scarify it with a cultivator, count the seeds that they had in a package of a certain species, put it on that exposed mineral soil, kind of blend those seeds in, cover it back up with the leaf litter, put a stake in it, make sure they gave us their initial data. We walked away for a year or so and came back to see how many seeds were establishing both in residual woodlands and post-agricultural woodlands. In 2016, we planted nearly 30,000 seeds in several different study sites. So to break this down, here's what we found from that. I've arranged these data by species. So we planted 2 baneberries, white and red baneberry, leek, blue cohosh, false Solomon seal, and red trillium. And what these bars are showing is the percentage, the proportion of the total number of seeds that we put on the ground in several different study sites that emerged and that we could count in the spring of 17, that's the following spring, the spring of 18, and the spring of 19. And so what we really thought was across the board that we'd see high establishment and germination the first year. Maybe many of these germinants would not make it through the drought of their first year and they die. And over a couple of years, we'd see the numbers reclining. And so you can see that in a couple of these clusters of bars. You can see high initial germination and then the establishment tailed off. It wasn't the case for all of them, but it was for a few. I've then organized this by each of these species between residual and secondary. Residual and secondary. The question is there something about secondary forests that inhibit them from having these plants establish? And so what you can see is, well, there's differences among species. The baneberries generally didn't do well. These other species generally did do well. There's a bar missing for blue cohosh from 2017. That's because, just FYI, blue cohosh has a double dormancy period. It did not germinate in the first year. We had to wait for 2 years out, 2 winters, 2 stratification periods for those seeds to germinate. And once they got that, they did quite well. The other thing to see here, though, is if you look at let's just focus on the 3rd year survivorship. If you look at the residual versus secondary, residual versus secondary, residual versus secondary, what you see is that in general, the residual forest and the secondary forest did just as well in terms of the proportion of seeds that persisted. And in fact, if there was a difference, the difference was actually in the direction of greater survivorship in the secondary forest in all of these species versus the primary residual forest. So the good news here is if you put the seeds on the ground, they grow. The curiosity is, I still don't know what's keeping them from growing naturally. Doesn't seem to be germination, doesn't seem to be dispersal, doesn't seem to be pollination. If you put them there, they grow. I still don't know what's keeping them from growing in these secondary woodlots. But what this is also showing us is that we have the potential for future restoration. All right. So maybe let me stop here. We're at close to the hour, I think. Kristen, Robert, I'm looking at you. Are we at the close of our time here, or do we have more? I think my time's about up. I want to leave some time for questions.
Greg, if you have a little bit more, we could do a couple more minutes, and then people could leave. I would say you could bring the talk pretty close to 1, and then folks can take off, or they can stick around for questions if they want.
Okay. Let me stop there. Maybe we can address some of these other things with questions, and if we need some more data, if some questions come up, we can come back to it. My bottom line that I wanted to get to with you is we have a need to restore forests in New York of their diversity. I'm very happy to hear participation and the interest of restoring understory forests up at Rice Creek. Anybody who has a woodlot in their backyard or has access to a woodlot or a municipal woodlot can engage in this type of restoration. I'm convinced that there's no limitation of pollinators and the ability of these plants to establish. There might be some interaction with putting plants out at small densities and not having pollinators find them. Ant dispersal does not seem to be an issue in the secondary forests. If you put the plants on the ground, they germinate and they seem to establish. I've learned that you can propagate these plants vegetatively and maybe get them to a reproductive age faster than putting 100 seeds on the ground, having 10% of them germinate, having half of those die, and then waiting 10 years for them to reach reproductive age. So sometimes maybe using vegetative material might be more effective. The problem is you end up depleting perhaps local sources of that during your restoration. There's an issue of managing light. What we're learning is if you open up the canopy a little bit, you get more flowers per plant, you get more seeds per flower. And by providing a little bit of energy, maybe we can speed up this restoration project. There's good potential for restoration. There's great opportunities for citizen scientists to do this. And the bottom line is now we go do it. Just go get it done. There's opportunities that abound in doing this. I'm happy to help anybody who might have an interest in pursuing this. So do let me stop there. And perhaps if there are any questions, we can visit about those.
Wonderful, thanks. If anyone has questions, feel free to unmute yourself and ask, or pop those in the chat. And we understand we're at the top of the hour, so at this point, anyone who doesn't have questions, you're welcome to leave. But we do have some time if anyone wants to stick around and ask questions, certainly.
And I'm happy to stick around. Yeah.
Awesome.
So I see Laura has a question. Does deer predation seem to be a problem? So yes and no. And there is evidence that deer are important dispersal mechanisms for some of these plants. For instance, trillium. A paper was done that showed that deer will eat the fruit, the berry of a trillium. I've tried this myself. In fact, they're quite sweet. They're mushy. The texture is awful, but they're not too bad. So I can see why deer might go for them. And in fact, they then will disperse the seeds. However, if deer populations are too high, more regularly what I see is the deer will be eating the flowers. And so if they're eating the flowers, they're not going to obviously have a chance to produce the fruits and the seeds. Sometimes they eat the entire plants also. So in certain locations, the deer populations are so high and the plant populations are so low that the deer are in fact having a negative influence on the ability to establish these plants. And some of the exclosure experiments that we've done along with this are showing us that. We don't do experiments without exclosures. But where the populations are lower, they in fact might be of value. So there's definitely a sweet spot. There's a sweet spot for maintaining diversity and regeneration and understory biomass and diversity of understory plants, but otherwise they can be detrimental. I don't know what that level is. I've not seen a good estimation of the deer densities that would have positive influences versus negative influences. But in general, I think in our central New York region, the deer densities are higher than they need to be and they should be. In fact, I go out, there's one place out back my back door. I go walk and see and enjoy the trilliums in May. And then by the beginning of July, I go back there again and there's not even a trillium leaf left. There's nothing but little popsicle sticks sticking up from the ground where they've nipped the plants right back. So they're definitely not having a positive influence in those forests. Good question. Well, it's really simple to do the restoration. I've done this in controlled conditions with undergraduate students. I've also done this as service projects. And what I've learned is that you can get a bunch of high school students, get them charged up with some candy bars and soda pop, give them a bunch of seeds and send them off into the woods to disperse seeds. When you do that, the problem is it's tough to monitor the outcomes of that, but you can at least start getting this going.
Do we have any other questions? Oh, there is one in the chat.
Do trillium and understory herbs need a higher pH? Let me just beg your forgiveness for a moment and share my screen again. And I'll go back to that one ordination. There. So the red and white trillium prefer higher pH soils. They require higher pH soils. The striped trillium or painted trillium, where is it here, occurs on more acid soils. So they actually separate based upon their degree of tolerance for soil acidity. You typically don't see painted trillium growing side by side with red and white trillium. Red and white trillium do require those higher pH soils. So depending on the site that you're on, you would utilize red and white or painted. And if you wanted to do this based upon the soil pH, you would have maybe different collections of plants that you would want to play with in order to do your restoration. Okay.
Greg, I have a question. So anyone interested in doing this restoration, do you have recommendations of where to obtain seeds or plants?
Right. So I don't have recommendations. There are some nurseries and seed suppliers that provide seed. I know that there are some local nurseries, and I would have to dig into my notes to give you specifics, that do this, and frankly do it, I think, in an ethical fashion also. What we don't want to be doing is completely depleting our forests of material in order to restore. And I'm not convinced that a lot of commercial operations do this in a sustainable fashion. The approach that I'm beginning to take, Kristen, is to begin to establish local excluded gardens, essentially do the captive breeding, and to gather the material locally, get it into an exclosure, use that exclosure to capture the seed, to not share the seed with wildlife, but in fact to put it on the ground and get it growing within that exclosure. Some of the work that I didn't show you is that it's possible to cut the rootstock and the rhizomes of these plants and essentially divide them and grow them, essentially fashion them from seed. So if you can do that and start the process of having a local dedicated source that you can go back to year and year and again, forgive me, I had the phone next to me, my apologies. To have that material year and again, you can go back to it and use it for a local source. What I have been doing though is going out and gathering seed, from woodlands around here and putting that seed from what I know is acid site onto acid sites and more base rich sites onto base rich sites. I do that carefully though. I really try not to take everything that's out there. Does that answer your question? I can get you some nurseries. Otherwise, I think it's just a matter of getting your own go-to garden established to keep going back to.
Awesome. Thanks. Well, I guess we're coming up on time, but I do want to mention that Dr. McGee there is a web page about ESF's Wildflower Restoration Project if people want more information. Do you have any other final thoughts about how folks could get involved, Greg?
Yeah, so first of all, that web page offers some guidance on maybe how to set up a restoration project. But if you look at the materials on the web page, they really are emphasizing monitoring and going back and monitoring your efforts. I really think I'm one of these people, there's nothing that I want to do in life without knowing what the outcomes are of those activities. And so if you go out and just scatter things, you don't keep track of how many seeds you put on and where they are, you don't really have a good idea of how effective you're being. So a lot of those materials that we're showing are simply some ideas about how to set up and organize, maybe systematic sample, partition the seeds, maybe put them in little clusters so that you can follow them through years, and you can keep track of your efforts through time. Otherwise, I think, where people go to do this is actually in their backyard. We've had a lot of success with local nature centers. So that Guppy property that I showed at the beginning of the presentation, that's actually municipal land for the town of Skaneateles. We've been partnering with the town to do restoration on a couple of plots there. Baltimore Woods is another nature center that's nearby me, that's west of Syracuse. They're doing this also. School groups are oftentimes looking for activities. I think you can partner with school groups. The idea would be just to be able to merge the landowner who wants to restore the land with the people who are willing and able to contribute and participate. So it really is as easy as that.
Wonderful. Well, thank you so much. It was wonderful to hear about this project, and we appreciate you taking the time to speak with us today. As Robert mentioned, we'll put a recording of this up on YouTube. If you enjoyed today's webinar, next week we are going to have folks, Kate Spector and John Mills from the Office of Sustainability, who are going to be talking about the project on our campus to reestablish the Centennial Arboretum. So that'll be neat. That's going to be both in person and over Zoom. So again, thank you, Dr. McGee. This was wonderful.
Well, you're welcome. Thank you for the invitation. It was great to visit. And Chris, and I would say that I see that Mary had a question. I'm happy to stick around after and answer Mary's question.
Sure.
Okay. Thank you for having me. It was wonderful to visit with y'all. You well.